Good evening, everyone. I hope you can hear me. So, we'll start with the questions for week 5 lecture today. Now, so the first question is gel filtration chromatography separates proteins based on dash. Four options are given here: charge, hydrophobicity, surface area, and affinity. So, for this question, you need to think about what property of protein is used in gel filtration chromatography. It has another name called as size exclusion chromatography. You can think about this question for a couple of minutes. Please do not put the right answer in the text box. Instead, what will be helpful is if you are able to eliminate the wrong options. So, whoever knows the answer, the right answer, can wait for a couple of minutes and then start with one wrong option that they think is incorrect. Okay, so Sai Sampath has given an answer, but yeah, before you give the right answer, what I think will be helpful is you can talk about one incorrect option. You can unmute if you want to talk about any incorrect option that you know of here.
The charge is incorrect, sir.
Charge is incorrect. So, in what type of chromatography do we rely on charge?
Ion exchange chromatography.
Okay, good. Okay. Do you want to talk about any other incorrect option as well?
For affinity, it is affinity chromatography, I think so.
Okay. What kind of affinity chromatography are you aware of?
Yeah, I don't remember that, sir.
Okay. Do you remember nickel NTA?
What, sir?
Do you remember nickel based affinity chromatography?
No, sir.
Oh, okay, okay. There's this one method called nickel NTA affinity chromatography where you have a protein with 6 histidines which forms a his tag, and this binds to nickel or cobalt. So your beads that you use for pulling your proteins will have these nickel or cobalt, and your protein with his tags would bind to that. So that is one example of affinity chromatography. And hydrophobicity. Do you know of how hydrophobicity can be exploited in chromatography?
Sir, it is HIC, right, sir?
Right. Like we looked at it last week, hydrophobic interaction chromatography, where salt
Salting
concentration.
in, yes.
Sorry? Yeah, you can go ahead.
Salting in and salting out. We are going to edit it.
Right. You basically change the salt concentration in such a way that it exposes the protein's hydrophobic regions, and then you change the concentration to elute it. Right. Good. Good. So the right answer for this question is, for gel filtration chromatography, we exploit the surface area. And can you justify why surface area in gel filtration? Any insights on that, if you know about it?
It consists of bits. So based on the size, it gets, elute. It takes longer time for smaller molecules, and larger molecule doesn't get inside the base, so it gets faster elution.
Right, right. So, to yeah, that is based on the size you're telling. Just one more additional point that we need to know is, we can consider, when we are trying to model these, we consider these as spheres, and spheres surface area is dependent on its radius. So the formula for the surface area is 4 pi r square. So because it is 4 pi r square, now if if the radius is larger, then surface area will also be larger, so the molecule's size will be larger. So merely based on that mathematical relation, we could say that here gel filtration chromatography is based on surface area. Like you had mentioned, if we have molecules of different sizes, some will go into your bead and have a different path which takes more time, and these are smaller particles, whereas larger particles do not get into the beads but rather go in the gaps between the beads, and they elute faster. This is the principle. The next question is, in the equation Vt is equal to Vg plus Vi plus Vo, Vo refers to dash. So there are four options for this: total volume, void volume, volume of the gel matrix, and pore volume. You can think about this question for a couple of minutes. This is These are four different terms that are used for volumes. And, to answer this question, first you can try to expand what these four different terms would mean. You can think for a couple of minutes, start with what these four different terms would mean, and then we can take it from there. Right. So Sai Sampath has told that VO refers to void volume. We'll go through what the different terms would mean as well. So here Vt is the total volume, Vg is the volume of the gel matrix. This is, in this particular case, this bead, the volume that is occupied by the beads. Vi is the internal pore volume, which is, so within the beads also your molecules can travel. So the volume that is within there, that is your Vi. And finally, Vo is the void volume, which is outside. So proteins that do not get into the bead go through the spaces between the bead, and this is the void volume. And they are asking for VO. So the right answer here is void volume. Good. The next question is the hydrodynamic radius of four proteins are as follows. W is equal to 4.65 nanometers, X is equal to 1.03 nanometers, Y is equal to 3.94 nanometers, Z is equal to 1.94 nanometers. The order of retention time for these proteins will be dash. Four different options are given here. So before we begin with for this particular question, whoever knows the answer can first tell us what hydrodynamic radius is and try to differentiate that from radius of gyration, because there are multiple different quantities that you can derive for a given protein, and we can assign it different radiuses, radii. So if we know the differences, it will be helpful. So whoever knows the meaning of hydrodynamic radius can unmute and tell us. So, does anyone want to try answering what hydrodynamic radius is and how to differentiate that from radius of gyration? Ritsia, Tara, Sai Sampath, anyone? The difference between radius of gyration and hydrodynamic radius?
Sure, I know about the hydrodynamic radius, but I don't know about the other one.
Okay, so what do you know about hydrodynamic radius?
It is nothing but the radius of the protein plus the hydration sphere which is formed around it.
So, what is a hydration sphere?
When the protein is dissolved in the liquid, if it
Okay.
is the hydrophobic, it forms a hydrophilic shell.
Can you repeat that again? Sorry.
When the protein is dissolved in the solvent like water, it
Right.
forms a hydration shell.
Yeah. So, does that mean that there are water molecules that are on the surface of the protein and it exceeds the normal protein's radius? Is that
Yes,
what you're
sir.
trying to say? Yeah.
Yes, sir.
Right. So if you consider any protein um, in aqueous solution, it would be surrounded with water molecules unless it's a membrane protein. There you will have this lipids. So I just have a figure here that would help us understand how exactly we can differentiate the different radii. So what they show here is for an example enzyme called lysozyme. So here they have constructed this. I mean, they've created this map from the crystal structure of lysozyme, and it is slightly elongated. It's not entirely spherical, and they've given it a molecular mass and an inverse density. So they say that the radius of gyration is where you have the mass and the distance from the center of the mass. So if for a protein which you can define in two axes and which is not equal, which means this is an ellipsoid. There is 45 angstrom length on one axis and 26 angstrom length on the other axis. So it's clearly not a sphere. Now, in this case, you have uh, a center of mass that is at the origin of the protein, the middlemost point. Now, the smallest sphere that you draw here that would cover this axis is your radius of gyration. So, with this, you will be able to tell approximately a radius, and that will tell you things about how this molecule can move. That is why radius of gyration is important. Whereas you have another thing called the hydrodynamic radius, which is given in blue here. So this is the radius which includes both the solvent and the shape, like what Sai Sampath had mentioned. So knowing the differences between these two helps you understand these phenomenon better. We use this term called radius of gyration when it comes to solution based experiments to approximate such a large particle as a sphere. So coming back to the question, so they have given the hydrodynamic radius of four proteins. Now they're asking for order of retention. So what we can approximately or what we can tell for sure is that if the hydrodynamic radius is large, the protein will be large. So the protein that would have the highest retention time, which means it will elude the last, will be the smallest, and so on. So the one with the smallest hydrodynamic radius, which is the smallest protein, is X, followed by Z, followed by Y, followed by W. So that is option C here. I hope this was clear. Okay. The next question is, the smaller protein molecules enter the dash part of the beads. In four options are given here: inner, outer, does not enter beads, and exterior. You can think about this question for a couple of minutes and then unmute and tell us the answer. Okay, so we saw earlier that small proteins enter into the beads, whereas large ones do not. So here the right answer is inner part of the beads. The next question is, the choice of matrix for a given gel filtration chromatographic experiment depends on the dash: fractionation range of the column, charge on the matrix, hydrophobicity of the matrix, and affinity of the matrix. So you can think about this question for a couple of minutes Okay, so in gel filtration chromatography, what we need to understand is our proteins can either enter the bead or not enter the bead. So whether our proteins enter the bead or not depends on the size of the protein, which means that we have various ranges for the size of the protein, which again means that we can have various ranges for the size of the pore as well. There are some proteins which forms a higher order oligomer that could extend in terms of megadaltons, whereas some proteins will be lesser than 100 kilodaltons. So depending on this, we have different columns that can allow different size range of proteins to enter. The same protein might enter the pore in one particular column, whereas go through the void volume in another particular column. So this is called as the fractionation range. And so the choice of the matrix for a particular experiment would depend on the fractionation range. So it basically depends on whether your protein, the size of your protein. Was that clear? Right. The next question is the molecular weight of a protein is 300 kilodalton. The same molecule gave two bands in an STS page, one of 90 kilodalton and the other of 180 kilodalton. The protein is a dash. question. How would you approach such a question? You're given the total molecular weight of a protein, and they say that you see two bands, one of 90 kilodalton, the other is 180 kilodalton. The protein is a dash. This is the question. How would you approach this? Yes. Does anyone want to try answering this? Okay. So one thing that you need to know is here the molecular weights do not match up. What they say is the total molecular weight is 300 kilodalton, but the two bands that they see is 90 kilodalton and 180 kilodalton. What could this probably mean? So, uh, one thing that this could probably mean is this is an anomalous run, which means what they see as 90 is not 90, what they see as 180 is not 180. But they're asking about the protein. Um, for sure, because we see two bands of different molecular weights, they are not of the same, the chain lengths are not similar or identical, which means that it's not a homodimer. Then what else could this be? It's a hetero. It's a heteromer, which means it has chains of different lengths. Now, will we call this a heterotrimer? No, because we see only two bands. Now, will this be a heterodimer? Possibly, because there are two bands. Then why is there a discrepancy in the molecular weight? This could be because in some cases you have post-translational modifications which can give you these anomalous runs, and depends on the salt concentration as well. But here, since they're asking only about the protein, we can conclude this is a heterodimer. Fine. The next question is the analyte with the KD value greater than 1 is dash, exploded from the pores, completely inside the pores, absorbed by the column, resolved at a lower volume. We can think about this question for a couple of minutes, and then we'll discuss what KD is and so on. KD tells us about the retention value. So it is the elution volume minus the void volume by the internal volume. And if KD is equal to 0, it means that elution volume is equal to void volume, which means your protein does not even enter the pore, it elutes in void volume. If KD is equal to 1, it means that your elution volume and the internal pore volume is equal. That is, your protein is within the pores. What they ask here is, what is the KD? What does it mean if KD is greater than 1? If KD is greater than 1, then elution volume is greater than the internal volume. So this would mean that the it is inside the pore, because KD cannot be greater than 1. Either it has to elute when the void volume is eluting, or it should be greater than your void volume, but it can not be greater than 1. The answer is, it is adsorbed by the column. Okay, next question. To regenerate the column for gel filtration chromatography, dash is passed through the column, and 4 options are given here: imidazolium, 6 molar guanidinium hydrochloride, salt solution, and then EDTA solution. Before we go on to discussing what the right answer for this question is, we need to know what is the role of the different components. So to start with, you can start by eliminating incorrect options. So whoever knows the right answer, first you can start discussing about one incorrect option. It could be any of the ones here, and then we'll go to the right option. Okay, so if we look at the options here, the first option that we can eliminate is guanidinium hydrochloride, because guanidinium hydrochloride can be used to denature proteins. It will remove any sticky proteins that is used, that is bound to the beads of your column. We can eliminate them. Then if you look at functionality, imidazole. Imidazole is something that we use as a competitive binding agent to be able to elute proteins that are bound to your nickel and TA beads, stacked proteins. EDTA, that is a chelating agent. It will remove your metal ions that are bound to your protein. So the only option that we are left with is salt solution. So these beads ensure that their size is retained in a particular concentration of salt. For example, 150 millimolar NaCl and so on. So for regeneration, we usually pass it through salt solutions. Fine. The next question is, to study the protein folding using gel filtration chromatography, the protein is treated with dash salt solution, imidazole buffer, glutathione buffer, and urea. Right. Why would you say urea, Sai Sampath?
So urea is a denaturing agent. It's unfair to protein structure.
But why would you need a denaturing agent here?
So since we are need to study at the protein folding, we use urea to know the structure, like it's primary or secondary or tertiary.
No, when you add urea, what happens to your protein?
It unfolds, I think so, sir. Get denatured.
Yes. So if it's denatured, when it goes through gel filtration chromatography, what would happen? What is expected?
Uh, I think it divides into like different. It gets broken down into like different proteins, like different sizes.
That doesn't happen. So your protein is basically covalently linked. You would have heard about amino acids that are linked by your amide bonds, right? Now, when you add urea, urea resembles amino acids. So the chemical formula for urea is NH2CO NH2. It has an amide group and a carboxyl group, similar to what you have in amino acids, because of which it can replace your amino acid interactions. So your secondary structure no longer exists. So now your protein is just a linear chain. Now, when you have these linear chain, you would get something equivalent to that of a denatured protein, like how it runs on STS page. Now, the thing here is, uh, when you treat it with urea and run it on gel filtration chromatography, it would enter the pores easily. It would mimic uh, the it would mimic chain like a polymer instead of a three-dimensional structure. So with that, you'll be able to, like you mentioned, again study protein folding. But how the protein, denatured protein, passes through your gel filtration chromatography can be studied with this. So this can be used as a comparison with respect to how your protein behaves in normal solution. That's the only difference that is expected. we look at other options such as imidazole buffer and glutathione buffer, these are not very relevant because this depends on what is the kind of affinity chromatography that you use. If you use a his tag, imidazole buffer would elute your protein. If you use a GST tag, which is glutathione as transferase, then that protein would elute. Whereas salt solution, now this is interesting because your protein can associate to form homo oligomers, that is homodimer, homotrimer, and so on, in salts of different concentration. So at higher salt concentration, like 500 millimolar NaCl and so on, your protein would not oligomerize. Whereas at lower concentration, like 50 millimolar NaCl and so on, at a given concentration, it could oligomerize into dimer, trimer, and tetramer. So in salt solution also, you can study the oligomeric status. But again, that has nothing to do with your protein folding, but rather arrangement of folded protein molecules. So that is why salt solution is incorrect here. Fine. The next question is, three proteins A, B, and C have similar molecular weights but different shapes. A is spherical, B is rod shaped, and C is randomly coiled. The order of radius of gyration of these proteins are dash, and four options are given here. Now, recall what we discussed about radius of gyration. I can show you that slide, and depending on that, think through that which of these uh, will have the highest radius of gyration, which of these have the lowest radius of gyration. This is the image for that. You can zoom in. Yes, you can go through what radius of gyration means here, and then accordingly try to tell us, depending on the surface shape of the molecule, which of these would have the highest and lowest. Okay, what we see here is your radius of gyration is the summation of mass of the atom into square of the distance from the center divided by summation of mass. So this would be an approximate sphere here. So for a protein that is spherical, the radius of gyration will be small, whereas a protein, yeah, so a protein that is spherical would have an intact radius of gyration, whereas in a rod shaped protein it will be uh, depending on one of the axes, and in a randomly coiled protein it will be very difficult to assign it because you don't have a clear distinction as because of the flexibility of the molecule. Now, depending on this, what will be the order? Now, because the protein that is spherical will have max maximal radius, so that will be the that will have the highest radius of gyration. That is why your RA will be the highest. Then here they say that randomly coiled will be greater than rod shaped, but that depends because like you see from here, at least here in your elongated protein you have such a radius of gyration, but in a randomly coiled protein it could very well not be aligned. Sorry. Okay. Okay, so one thing that I couldn't mention here is these random coils would mean loops that are connecting distinct domains. You have secondary structures that are connected by loops, and random coils would lack this structure, so it will be disordered. So that could possibly explain that it takes multiple conformations, because of which it would have a higher radius of gyration compared to your rod shape. That is why we go with this option, that your spherical protein has highest radius of gyration followed by random coil followed by your rod shape. Fine. The next question is, dash is used for making the stationary phase of a gel in a gel filtration chromatographic column, and four options are given here: agarose, dextrin beads, resin beads, and carbon nanoparticles. Okay, so for stationary phase of the gel, we can either use agarose agarose or textile beads, we will not be able to use resin beads. Resin beads are nothing but these that contain your nickel NTA and so on, and definitely not carbon nanoparticles. So depending on what is used, we either get Cephadex if it's textile and Cepharose if it is agarose. These are the two common kinds of columns that you observe that people use for protein gel filtration methods. The next question is, to obtain a CD of purified protein in ammonium sulfate buffer, we need to perform dash: phenol chloroform extraction, desalting, imidazole treatment, and urea treatment. Okay, so we need to understand what this ammonium sulfate buffer is. If our protein is not is tagged, then what we can do is, when we grow these cultures and pellet it, we can lyse it and precipitate our protein with different concentrations of ammonium sulfate. So ammonium sulfate is widely used to precipitate proteins, and depending on the nature of our protein, it will get precipitated or solubilized at different saturation levels of ammonium sulfate. People generally use 30 to 90% ammonium sulfate to saturate. So, if you have your protein in a solution in a buffer, if you have your pellet, let's say you grow your cells containing protein, and these are pelleted. Now you will resuspend this into a buffer containing uh, some necessary salts and reducing agent and glycerol if needed. And what you would do is, you would add a few grams of ammonium sulfate to ensure that in this solution you have saturating concentrations of ammonium sulfate. So you can decide either 30%, 60%, or 90%. Now what you would do is, you would add it, stir it, and then you would centrifuge your solution. So at a particular concentration, for example 30%, more than half the cytosolic proteins in E. coli would be uh, precipitated. So when you run this on gel, you would know in which concentration your protein of interest would be precipitated. This is where ammonium sulfate buffer role comes. Now, for CD, you can't use this because CD is a very a sensitive experiment that determines the secondary structure of your protein, and the measurement is made between 190 till 230 nanometers wavelength. So you cannot really use any salts that would absorb in that particular range, which means that the ammonium sulfate has to be removed. So for removing that, we perform this process called desalting. This can be done in two methods. One, using dialysis bags, where you have an external solution of zero concentration of ammonium sulfate. So because of dialysis, the salts get exchanged and your protein is now free of any of these salts. The other is a method similar to your gel filtration chromatography, where you pass in your protein through a column, but instead of getting into pores, your protein enters through the and exits through the void volume, and your imidazole or your ammonium sulfate will be removed. But generally, to preserve the nature of the column, people do not use uh, your desalting columns for when it is done with ammonium sulfate. although this method is called desalting. So the right answer here is desalting, option B. The next question is, if a protein is incubated with 8 molar urea for a long period of time, it will be eluted with dash. And 4 different options are given here: Vg, Vo, Vi and Vt. We earlier saw what these different terms are. So you can think about this question for a couple of minutes and then we'll get back to what happens when you denature your protein and run it through a gel filtration column. Okay, so we earlier saw this. VO is the void volume, VI is the internal pore volume, VT is the total bed volume. Now, when the protein is globular, it can either enter through your internal pore volume, or if it's very large, it will elute through your void volume. But if it's denatured, it will become a random coil. So that is equivalent to your smaller molecule. And they say that it can penetrate the pore, so it will elute at the total column volume, which means it will go through the entire length of the protein into all the pores, take a lot of time, and exits last. So the option is your VT. The next one is, to purify proteins based on molecular weight, dash is used. So one another name given to gel filtration chromatography is size exclusion chromatography, and that is mentioned here. And the last question for today is, a protein is treated with 2 molar and 8 molar urea. The retention time of different species in increasing order would be dash. So what they're asking is, when you have 2 molar and 8 molar urea, which is different concentrations, and that would mean different unfolded forms, partially unfolded and fully unfolded form. So the retention time in increasing order would be dash. So your native protein will have a particular retention time. A fully unfolded protein will have a particular retention time, and your partially unfolded protein will be in between these. So if we go according to that, either option A or option C is the right one. Now they ask for retention time of the different species in increasing order. So option so the third one here should have the highest retention time, which means it will elute the fast. So based on this, we go with option C. Native protein greater than partially unfolded protein greater than unfolded protein. The unfolded protein will go through all the pores, take the largest route, and will elute last. Right. So with this, we come to an end of this week's set of questions. If you have any questions, you can ask here. I'll be staying on the call.